Now that we've learned the characteristics of both prokaryotic and eukaryotic cells, let's talk about some of the specific organelles that you would find in eukaryotic cells. Now, this video can sometimes run long. I'm going to try not to talk very long because it's basically just a laundry list of all the things in the cell and what they do. If you need any more information, your book has a ton of information on more specifically what each of these organelles does in the cell. So the largest organelle in the cell is the nucleus. The nucleus is important because that's where all of our DNA is housed. Now remember that DNA is our genetic material, which means that it is all the instructions for how our cell is going to grow, how it's going to develop, and all of the instructions for the proteins that are going to be made that are going to be doing all of the jobs that the cell does. So the DNA contains all of the instructions for that cell to perform all of its living functions. The nucleus is surrounded by a nuclear envelope. This is a membrane that surrounds the nucleus and isolates it from everything else in the cell. Now, this is different from a prokaryote where we have a nucleoid, which is basically just a region where the DNA hangs out. It's not separated by a specific membrane. The DNA always stays in the nucleus. It never leaves. And this is important because we need to get that information out of the DNA, but we also need to protect that DNA so that that DNA can be used for a long time as it refers to the lifespan of the cell. And so the nucleus not only houses it, but maintains that information. Now, the nucleus also has pores. You can see them here in this image. And those pores help to regulate movement of protein and RNA. So some things will move in and out of the cell to get to the DNA. Now, proteins do all the work in our cells. And we have a combination of proteins and RNA that go into the nucleus, that copy our DNA, that copy it into RNA that can be used as instructions later outside of the nucleus to build proteins. So in our nucleus, we have maintaining DNA, we also can replicate the DNA, and that's important when we get into cell division. And we can also transcribe the DNA. Now, transcription is the first step in the process of gene expression, where we're going to take some information out of DNA, turn it into a little RNA message that can be used in the cell. And we'll get to that later. So inside the nucleus, we have chromosomes. Now, a chromosome is just the name of a single piece of DNA. That DNA can be linear in eukaryotes or circular in prokaryotes. Now, obviously, if it's a prokaryote, it's not in a nucleus, but we still call it a chromosome. It's just a long piece of DNA. And that really long DNA has to be coiled very specifically so that it all fits into the cell. Now, it is coiled into a structure called chromatin. So chromatin is what we call chromosomes that are folded and condensed around proteins. There's a little picture down here in the corner. This blue, if you can see the blue there, that blue strand is the DNA, and these little purple spiky guys are the proteins that the DNA is wrapped around, and they help to compact it in a very precise way. Also inside the nucleus, we have the nucleolus. This is a region within the nucleus where we produce RNA. And we produce a specific type of RNA here called ribosomal RNA. If you remember, one of the components of all cell types are ribosomes that make the proteins. And ribosomes are made up in part of this ribosomal RNA. So if we're going to express our genes from our nucleus, we need to have this ribosomal RNA. The structure that actually does the protein production, that is sort of the intermediate between our DNA encoded information and our protein, where we see the outcome of the DNA instructions performed, is the ribosome. So the ribosome is essentially a workbench that builds proteins. Now, ribosomes themselves are made up of RNA called ribosomal RNA and proteins. There is a cartoon picture of a ribosome down here at the bottom. We will talk more about the structure and actual function of ribosomes when we get to chapter 14. But for now, you can just think of them as protein factories. They put together our proteins using the information that is stored within our DNA. So ribosomes are critical in the process of gene expression. There are ribosomes in two different places. Some ribosomes are bound to a structure called the Endoplasmic Reticulum, or the rough ER. That Endoplasmic Reticulum has ribosomes that are actually embedded. You can also find ribosomes free floating in the cytosol or cytoplasm, and you can see both of those depicted on this cartoon image here. We've got ribosomes in the cytoplasm, and we've got ribosomes on that rough ER. Most of the organelles that are found in the cell are part of what we call the endomembrane system. Endo, the word endo, means inside. So this is a system of membranes, plasma membranes, that is found inside the cell. Now, the endomembrane system's function is to move products, we're mostly worried about proteins, through the endomembrane system, process those products, and eventually release them into the cell or out of the cell, depending on where they need to go. The endomembrane system includes the nuclear envelope that surrounds the nucleus. I should say it encloses the nucleus, because the endoplasmic reticulum is found surrounding the nucleus on the outside of the nuclear envelope. There are two types of endoplasmic reticulums. There's the smooth and there's the rough, and they have slightly different functions. We also have the Golgi apparatus, which comes further outside that endoplasmic reticulum, and then lysosomes that are found outside of the membrane, sort of disassociated but still part of it. And then we also have the plasma membrane. It's not really part of the endomembrane system, but the endomembrane system can be built from the plasma membrane. Or I should say builds the endomembrane system. So the plasma membrane is not necessarily always contiguous, but it is kind of part of it, because products can flow between the rest of the system and the plasma membrane. So that endomembrane system is basically a contiguous system that is connected either because they are actually physically connected or by vesicles. Now, vesicles are essentially little pockets of membrane, and we'll see what some of their functions are in just a minute. So our endomembrane system is made up of the nucleus surrounded by the nuclear envelope, and then the first structure surrounding that nucleus is the endoplasmic reticulum. So if you look at this cartoon drawing here, we've got the nucleus in the middle, and you can see that the cutaway shows that the endoplasmic reticulum surrounds that nucleus. It is a large portion of the total membrane that is found in eukaryotic cells, and it is thought to have evolved by invagination or pinching in of the plasma membrane. So this entire system evolved that way. There are two types of endoplasmic reticulum, and the overall shape of the endoplasmic reticulum is either tubes, you can see those here, or flat sacs. So I kind of think that the endoplasmic reticulum looks like coral. If you look at pictures of coral, you have tube coral and you have sort of these flat shaped coral. That's kind of how I remember which part is the endoplasmic reticulum. But the endoplasmic reticulum consists of these tubes and sacs that are called cisternae. So if you know what the word cistern is, it can help you remember what cisternae are. Cisterns are basically jars that hold water. So these are sacs or tubes, not jars, that hold cellular products. So for example, we might make a messenger RNA in our nucleus, and it's going to come out and into the endoplasmic reticulum, into one of these sacs where it can be made into a protein. And then it can be transported through by basically transfer through the membrane of the endoplasmic reticulum. So there are two types of endoplasmic reticulum. The smooth ER are the tube-shaped sections of the endoplasmic reticulum, and its function is related to metabolic processes. So in the smooth ER, we get synthesis of lipids, we get metabolism of carbohydrates, so processing of carbohydrates, whether we're building them or breaking them. Detoxification of drugs, and what I mean by this is if you are in a system that is taking in foreign chemicals, products, things like that, it'll actually break down some of those products so that they don't accumulate in the cell. Little piece of interesting knowledge, the liver, which is important in the breakdown of alcohol, is high in smooth ER because it's breaking down a lot of those chemicals that are not supposed to be in the cell. The smooth ER also does some storage activities, including storage of calcium ions. The rough ER is the sort of pleated sac-like shape. And the rough ER is called that because ribosomes are found on the surface. So that should give us a hint as to its function, which is the production of proteins. So those ribosomes that are embedded on the rough ER are building up proteins. Remember, we can find ribosomes in two places, on the rough ER and free floating in the cytoplasm. Usually the proteins that are built on the rough ER are going to leave the cell, and we'll talk about why that is in just a minute. Products that are made from the rough ER, like our proteins, are then transferred or transported by vesicles. You can also have vesicle transport proteins out of the smooth ER. Those vesicles are going to go to the next structure in our system, which is called the Golgi Apparatus. So here's a diagram of kind of a blow up of our rough and smooth ER. Again, we've got the tube-like smooth ER, the rough ER. This picture is a little bit bigger. You can actually see a little vesicle pinching off. So again, a vesicle is like a little packet. It's basically like a membrane bubble that has something inside that needs to be moved around a cell. I like to think of it as like a delivery truck, but it pinches off of the endoplasmic reticulum, or it could pinch off the Golgi apparatus, it could pinch off a lysosome, it could pinch off or pinch into the plasma membrane. Those vesicles are just little pockets. So our cellular product is produced in the smooth or rough ER, depending on what we're doing. And then that little vesicle, that transport vesicle, pinches off and moves its product wherever it needs to go. In a minute, we're going to talk about the cytoskeleton. And that is basically a scaffolding that provides structure in the cell. And those little vesicles will move along this cytoskeleton track to wherever they need to go. So from that rough ER or smooth ER, and you can see the ER up here around the nucleus and then our Golgi apparatus, we move, those vesicles physically move, they pinch off of the rough ER or the smooth ER and move to the Golgi apparatus where they fuse with the Golgi. So here we've got this little vesicle that's come off of the ER. The side of the Golgi apparatus that's closest to the ER is called the cis face. Cis means same. You might recall that from our chemistry chapter. So it's on the same side as the endoplasmic reticulum. And the Golgi apparatus basically does processing. It processes the molecules that come into it. So one of the things that it might do is add a little molecular tag so that the cell knows what that is. As this processing continues, the product is going to move through the Golgi and eventually pinch off. Got that pinching starting here, pinch off into a little vesicle where we can ship out our finished product to wherever it needs to go, whether that's somewhere else in the cell or the plasma membrane. It leaves from the trans side. So trans means, you could say like opposite or different. The opposite side away from the rough ER. So in our Golgi apparatus, as our products move through the Golgi, those ER products are going to be modified. They might be stored for a bit, but they're basically going to be finished off so that the final product is ready to go once that vesicle is ready to leave. Once that vesicle leaves the Golgi apparatus, again, it travels along those little cytoskeleton tracks and it fuses with whatever membrane it needs to fuse with. So generally, this is going to be the plasma membrane. If it's coming off of the ER and the Golgi, it's probably going to the plasma membrane where it's going to be fused with the plasma membrane and the products are going to exit. So the finished product will actually leave the cell by fusion of that vesicle to the plasma membrane. The last structure that's part of our endomembrane system are the lysosomes. You can think of the lysosomes as like the garbage disposal. I also like to think of it as the stomach or the intestines. This is an organelle that is going to digest things in the cell. So this might be a compartment that is digesting food particles. So if the cell brings in something that it's intending to eat, to consume for energy, it might digest that. So it's going to digest the cellular macromolecules, just like you digest those molecules that come into your stomach. Lysosomes are also part of a process called phagocytosis. So in phagocytosis, the cell will essentially wrap itself around another cell and pull the contents in. So when that happens, a vesicle sort of pinches off from the plasma membrane. Once the plasma membrane has surrounded, then you've got a big vesicle that's full of stuff that you want to eat. The lysosome can also perform something called autophagy. So auto means self, phage means eat. So you can roughly translate this to eating oneself, but that's not really what we mean. Autophagy is where the cell will break down components that are no longer usable. Maybe they are worn out proteins that need to be disposed of. Maybe something, the cell's been damaged and there's a part that needs to be broken down and essentially broken into its components and gotten rid of. All right, so the last part of the endomembrane system is the plasma membrane. We've talked about this several times already. I'm not going to go into anything else. We'll talk about it in chapter 5. It's the structure that holds the cell together, and it regulates what moves in and what moves out. All right, these last few slides are other organelles. So the first is a vacuole. A vacuole is a storage compartment that has a specific function. So these can be found in plants or animal cells. They are often food vacuoles, so storage of food particles. They may be what we call a contractile vacuole. So a contractile vacuole is a section of the cell that can increase or decrease in size depending on the needs of the cell. So some organisms will regulate the water uptake using a contractile vacuole. So regulate kind of how much water is in the cell. That should not be confused with a central vacuole, which is a water storage organelle in plants. So in plants, the largest area in the cell is actually the central vacuole where water is stored. Now the central vacuole is important because that water storage gives the plant cell its fullness. So when the vacuole is full, you get a nice, beautiful, rigid plant. When the vacuoles in the plant start to become dried out, maybe they're not full of water, that's when you get wilting plant cells and wilting plants. So that central vacuole is really important in also regulating the storage of water and the shape of the plant cell. So if your plants are wilting, they probably don't have enough water in their storage vacuole and you need to water them. All right, so our last set of organelles are the energy transformers. So these are the organelles that have something to do with bringing in and converting energy into a usable source. The first one we're going to talk about is the mitochondria. The number one answer I get when I ask students of all questions that I ask, the number one most answered question is, what does the mitochondria do? And most of you probably already know, and you would say it is the powerhouse of the cell. And I'm going to tell you that on a test, that's the wrong answer. And what I mean by that, not that it's actually wrong, but I want you to tell me what it actually does. What does it mean to be the powerhouse of the cell? Well, in all eukaryotic cells, that means it is the site of cellular respiration. So cellular respiration is kind of analogous to respiration that you do, which is breathing in and out. It is taking oxygen into the cell, and that oxygen is used to produce energy by breaking down fats and sugars. So we get we get our energy stored in sugars or fats, whether we eat them in our diet if we're if we're animals, or absorb them from the ground if we're fungus, or if we collect it from the sun and convert it ourselves. But Those sugars and fats mean nothing if you can't break them down. So the mitochondria uses oxygen as part of the chemical process to break down those sugars and produce energy that can be used in the cell. Because sugar can't be used directly, we have to convert it into our energy currency, which is a molecule called ATP. So that is the function of the mitochondria. So our book shows mitochondria as these orange structures here. Really important in a few minutes, mitochondria have their own DNA. They have their own ribosomes, and that DNA and ribosomes work to express the genes that help build the mitochondria and also help the mitochondria to perform its function. The mitochondria and chloroplast that we're going to talk about in a minute are the only organelles besides the nucleus that contain DNA. That DNA reproduces separately from the cellular DNA, from the nuclear DNA. And in the mitochondria, we call it mitochondrial DNA. You might have heard the term mitochondrial DNA because it's often referenced when we are talking about human evolutionary relationships, because mitochondrial DNA is passed directly from mother to child. When reproduction happens, the sperm basically only sends in its DNA, whereas all the cellular components that were in the egg become the cellular components of that fertilized cell. And so the mitochondria that's passed on also passes on its DNA. And so to a limited extent, you can look at a historical and ancestral lineage through mitochondrial DNA, which is kind of cool. Mitochondria have special folds inside called cristae. These cristae are folds that increase the surface area so that we can get more energy production. We'll talk about that more in chapter 7. Our second energy transformer are the chloroplasts. These are found in anything that performs photosynthesis, so not in animals. Students like to come up with weird examples. There are some strange examples where there's a symbiotic relationship between an animal and a cell that performs photosynthesis, but they are not found in animal cells in general. They are found in plant cells or protists such as algae that can perform photosynthesis. So the process of photosynthesis takes sunlight energy and converts it into chemical energy in the form of sugar, specifically glucose. So this is really important because all of our energy comes from the sun. Oops, wrong kind of sun. From the sun. And that energy is taken in and converted into a usable form for cells by the chloroplasts. The chloroplast has a pigment in it called chlorophyll that gives it its green color, and the chlorophyll is found all along the inside structures of that chloroplast. Those structures are called thylakoids. Just like the cristae in the mitochondria, those thylakoids are highly pleated to increase the surface area of the sites for photosynthesis, so we can do more photosynthesis in a smaller space.